Hi everyone, I'm Gloria. I work in Coleman's lab at UIUC, and today I'm going to be talking about a quantitative characterization of a transposable element response function. In general, what we do in our lab is we use molecular biology and fluorescent microscopy to observe transposable elements inside living cells and then apply theoretical analysis to quantify that behavior. So the first thing you might be asking is, what is a transposable element? A transposable element, or TEs, are also colloquially known as jumping genes, so they can spontaneously move around within the host genome. They're found in all domains of life and proliferate over time. So due to their prevalence, they're thought to be a major source of genomic plasticity driving evolution. So if we can kind of understand how they're behaving inside a cell, then we can understand evolution at this genetic scale, which is pretty cool. So I'm going to walk you through the mechanism of transposition for the IS608 transposable element, and this is the TE that we actually use in our experiments. So this is what it looks like. So you have a sequence for a transposase, which is the protein that facilitates transposition, and it's flanked on either side by sequences known as the right end and left end imperfect palindromic repeats. So during DNA replication, the transposase is transcribed or produced, and as the replication fork passes the transposable element, if the right end and left end palindromic repeats are located in the lagging strand of the DNA, these hairpin structures form, and that's actually what the transposase recognizes. So it binds there and then excises. Through some search mechanism, the whole thing looks for another place inside the chromosome to reintegrate. So that's the mechanism. So what we did is we took this IS608 transposable element and then tagged it so that we could actually see these different processes happening in real time. So this is what our construct looks like. So you'll notice the first thing we did is we placed the transposase under the control of an inducible promoter. And what this allows us to do is it allows us to tell it to produce transposase and also how much. We've also tagged the transposase with Venus, which is a fluorescent yellow reporter, and we've inserted the entire sequence into a promoter region for M. cerulean, which is a bright blue fluorescent protein. So now, if we induce the cell to express transposase, it will fluoresce yellow because it's tagged with Venus. And if an excision event occurs, the first excision event, the promoter region is going to rejoin, and so we'll be able to see that because it will fluoresce blue as well. So the question we can answer now with this construct is, how does TE activity, or more specifically excision events, change as a function of transposase concentration? So to answer that, we take our transposable element, place it inside a low copy number plasmid, transform that into wild-type E. coli, and then grow up different cultures, each of them with a different concentration of inducer, which should correspond to a different concentration of transposase produced, right? And so, this is what you might expect to happen. The more inducer you have, the more transposase, and the more blue, because the more excision events, right? We expect that at some high inducer concentration that there will be a maximum number of excision events, and so there should be some maximum blue fluorescence. We made a control with just the M. cerulean inside the plasmids to control to see how high this maximum blue fluorescence is. And so these are examples of the images that we take, and so you can see for a single cell, by simply comparing how yellow it is to how blue it is, we can get a measure of how many excision events have happened per transposase concentration. So the first thing we did is we looked at the M. cerulean only control and found the mean maximum blue fluorescence. The second thing we looked at was a strain where the transposable element was inserted into the lagging strand. And that's what our data looks like. We found the mean of that also. And we're just going to be looking at this middle portion. Anything above, we didn't have enough data. Anything below, 1 nanomolar corresponds to less than 1 transposase molecule per cell, which isn't very meaningful. So here it is, zoomed in. Another strain we looked at was where the transposable element was inserted such that the right end and left end imperfect palindromic repeats were in the leading strand. And you see that we actually get different behavior. And so the reason for that is if we go back to how the transposase recognizes the transposable element, what it sees are these hairpin structures. And these hairpin structures can only form when the palindromic repeats are in the lagging strand. So if they're in, instead, the leading strand, transposase will recognize the transposable element as easily, and so we should expect less excision events for any given transposase concentration, which is what we see. Another thing you guys might notice is that there seem to be 2 distinct regions in the lagging strand of excision rates. So there's kind of this initial very fast increase, followed by a more gradual increase. We think this has something to do with the fact that transposase is a dimer, and so, as the concentration increases, it might start interfering with itself as it binds to the hairpin structures. But this is all very new data, so right now we're developing theories to give us insight into the behavior. By matching our theories to the data, we hope to be able to extract binding modes and parameters for the transposase as it recognizes the transposable element, as well as figure out how the excision and integration probabilities depend on transposase concentration. So in conclusion, we can detect excision events and transposase levels in live cells in real time with really high resolution. We're using that to look at how transposition probability, excision, and integration rates depend on transposase concentration. And in the future, we hope to be able to look at these cells inside microfluidics to see how the fitness of the cells are affected by transposable element activity, as well as look at how different environmental stresses affect the transposable element behavior. I'd like to thank everyone in the Coleman lab, as well as Nigel Lowenfeld and his student Mikhail for helping us with the theory. Thank you.
Questions from the audience? Let's hear from the students. Don't make me call on you.
Don't call on you.
So can you comment on how fret between the cerulean and Venus and folding maturity would affect what you're measuring, or how you include that in the model? Fret, you can get energy transfer if they're close enough, right? I mean, it looked like from the sequence, I don't know how far apart, but if you, from your diagram, if they were close together, you could actually get, if you get, I mean, you have okay, I'm sorry. So they're 2 separate genes, right? So you have the M cerulean and you have the Venus construct.
Yeah.
His question is, do you see fret between those 2?
I mean, are they being...
Energy transfer, that's
right.
what fret means?
Yeah.
That, I don't know. I'm sorry. This data we got just before I came here, so
okay.
We're still trying to develop a theory.
But you also did this model, right? And in the modeling, you need to, do you worry about maturation? How well the proteins maturate? Not all the proteins fluoresce in the end.
You're talking about this one?
That looks like it.
This one?
Yes.
Okay. Sorry. That.
Right.
Okay.
I mean, because in the end, you're looking at your intensity of the M cerulean to Venus, right?
Yes.
And then there's a question, I mean, do they all fold? And they certainly don't all fold. Whether that's a problem, whether you have fret that decreases the intensity of the M cerulean. We can talk later.
Any other questions from the audience?
Did you look at the dynamics of how long you're inducing the transposase, or I didn't quite get that?
Right, so the experiment that we did, we grew all of the different cultures for the same amount of time and just looked at them all at the same time point. We're also actively trying to look at how they grow over time, so this is where the microfluidics would come into effect. And videos like where this image comes from, where we're growing them over a long time and observing how you can see different cells are kind of growing or fluorescing different amounts, and we think this has to do with the transposable element activity. Yeah, so we are looking into time as well.
Okay, let's thank Gloria again.